So thank you. As Michael said, my name is Kathy Sokas. I am the global regulatory lead for several cell therapies within Janssen R&D. I'm also the chair for the Alliance for Regenerative Medicine Regulatory Committee. And with that, I get the privilege of working with a lot of great industry leaders throughout various organizations. So today, just to set it up before we introduce the panel, we know in today's regenerative medicine space that we've made a lot of progress, but there is still a long way to go for us to get to the mature paradigm from a regulatory perspective where this industry will be in years to come. There's challenges that we need to face, and we're doing that both from an individual level, individual companies, but also from an industry perspective. So ARM is one of those industry groups that is really trying to work with and address some of the topics that we deal with every day. And specifically the regulatory committee, as Michael mentioned, Ellen and I, we recently published a paper on FDA sponsor interactions. We have met with CBER on several different occasions, an ARM delegation, and that's good because it allows us to dialogue with the agency and to address potential opportunities where we can work together, both from the government perspective, but also from industry. And we routinely comment on guidances and regulations that are applicable to our field. We are working on potential workshops for global regulators. So we're active, and we really are hoping that this benefits the industry as we move forward. So today's panel, and I'll let them introduce themselves, but this will provide people with an individual perspective from companies at various stages of the development framework. So if the panel could each introduce themselves, that would be helpful for the audience.
My name is Ellen Feigl. I'm Senior Vice President of Research and Development at the California Institute for Regenerative Medicine. So one of my main goals really at SCMOM is to try and advance the science towards and into clinical trials. So we have a lot of challenges and also opportunities in trying to go down the regulatory pathway to develop a therapeutic product. And what we're trying to do, in addition to the funding, is actually working with the agency on ways to actually help facilitate and help better position our applicants to be better able to move down that pathway. So background, I'm a hematologist oncologist by training. I've worked in the federal government for 12 years at the National Institutes of Health. I've worked in small pharma as a chief medical officer in a large biotech in medical development. And in addition, I've worked at nonprofits and academic institutions. But always the focus has been on trying to move innovative therapies towards patients.
Hi, Karen Abudi, Associate Professor at City of Hope and CSO of Therabiologics and a SCMOM Disease Team Awardee. My INDs so far have going in through academic institute, through City of Hope, so it's a little bit different than applying through industry, but we'll be slowly transitioning that to industry.
I'm Patrick Bilbao. I'm Vice President of Regulatory Affairs and Government Relations for Organogenesis, and I bring the perspective to the panel of a company that has two cell-based products on the market now. As Jeff McKay described yesterday, we have our second product, Intuit, that was approved through BLA process earlier this year. And when you have products on the market, there are new issues that arise that you don't see in the development stage. We now have two sales forces, approximately 200 sales representatives. We're in a competitive environment, and some issues related to promotional activities, claims, and regulation of tissue-based products come about for us.
So I'm Ed Feigl. I'm the Chief Operating Officer of a company called Cytomedics. For those of you who didn't hear the presentation yesterday, we're actually kind of a unique company because we sit across two areas. We have point-of-care devices that are sold for regenerative medicine applications, and we also have cellular therapies that are heading down a BLA path for approval. And I'll probably speak more to the cell therapy side. In that area, we have faced a number of challenges. We've gotten through 7 INDs with our technology. We've actually gone and gotten a special protocol assessment, or SPA, with our products. We've actually gone through the potency assay issue with FDA and gotten one approved for a phase 3. We've gotten orphan drug status. So we've been through a number of the development-oriented hurdles from a regulatory perspective.
I'm Tim Fong. I'm Vice President of Technology and Process and Product Development at PCT. I've been in the drug development field now for almost 18 years, working primarily in cell and gene therapy as well as biologics. Currently, my role at PCT is to help our clients, both our parent company, Neostem, as well as external clients, develop their products from bench or bringing it out of the university in terms of bench science all the way through clinical manufacturing and also clinical manufacturing.
Thank you.
Commercial
I'm
manufacturing.
going to start off the panel with a broad question and ask each of them to respond. But if there's questions during the discussion, please raise your hand so it can be interactive and we can get some open and informative dialogue. So I just wanted to start off, and the broad question is, so what's the main regulatory hurdle that you've dealt with in your individual company, and how did you actually address that? And I know there might be more than one, but just if you could pick one or two to speak about.
Sure. I think, in our experience, it can be challenging for cell-based products to identify the mechanism of action and related to that, obviously, develop appropriate potency assays. It's something that the RM Science and Technology Committee is actively working on. And there is some guidance from FDA, but I think that's an area that can be improved, hopefully with collaboration with FDA. But when you're dealing with products, most cell-based products are multimodal. They work by several mechanisms of action. Potentially, it can be hard to define the exact mode for the clinical benefit of the product. And for instance, our Gentuit product contains 2 cell types. It contains a collagen matrix, and it obviously has living cells, but also produces many cytokines and growth factors, has a physical barrier as well. So through the BLA process and the clinical stage process, we had to do a lot of work in characterizing the product to identify potential modes of action, and doing that both with in vitro assays, preclinical, in vivo work, and really trying to narrow down before we get into clinical trials or late stage clinical trials what might be the best potency assay. But we didn't fully define the potency assay or get agreement with FDA until the end of really the BLA process. And we have a post-marketing commitment to generate further data to demonstrate that the potency assays that we have developed are reproducible. So ideally, you can do that earlier, try and do that during phase 3 clinical trials, but I think it's important to get to FDA early on to present a characterization panel to them of the potential modes of action of your product, and generate data where you can show that potency assays are reproducible, specific, and sensitive.
So Patrick, did you actually communicate with the agency proactively, or what did it come out about when you submitted certain IND submissions and they would respond to?
Yeah, I mean, we had some discussion certainly before the BLA phase, but it really wasn't until we were able to present more data, more robust data in the BLA, and through our discussions with them. We had several rounds with the scientific review team regarding the potency assay for Gentuit. So, as I said, it really wasn't until the end of the process when we fully defined what the potency tests, and it's multiple tests, a panel of tests, would be for the commercial product, again with a post-market commitment to present additional data to the agency.
I agree with Pat. I think cell characterization assays, particularly with potency, is an important issue for us, particularly for those people early in development. We often get clients that come to us with literally a 2-pager protocol saying, design a GMP process for manufacturing my product for a clinical trial. And so I think these assays are critically important, because as you make process changes or improvements, you want to make sure that you're not changing the character of your cell product, and more importantly, the activity of your cells. So I think a lot of the developers really need to think ahead a little bit in understanding what their final product concept is, and then begin to build those assays and specifications around their final product, so that as you go through the process development and product development, you can be assured that whatever you're changing in your process for improving yield, improving viability, or whatever other purpose, that you're not altering your product at the end. So I think that is a big challenge in development.
I think, the Office of Cell Therapy recognizes the complexity of these types of products, and I think their answer to that is to have more than one potency assay, in many cases, a panel that looks at different aspects of the product and ties it to reproducible biological function and not necessarily the clinical function of the product. That's the most difficult challenge, is tying it to the clinical effect of the product.
So Ed, did you find the same challenge? You had different challenges in your development.
On the potency assay?
Yeah.
We were lucky because we were in the cord blood area, and fortunately there's a lot of data there that correlates CFUs with engraftment, and then we were able to correlate our cell population's activities to CFUs, and so that was a very nice chain in order to make that argument on potency. So but that's the easy path, right? I mean, that's an easy one, right? But if you get into cardiovascular, where those kinds of assays are not as well known, then that's a much more difficult story, I think.
So Karen, from your early perspective?
So the additional things that we had, if you're working with an allogeneic line, you also have the immunogenicity and the safety studies that need a lot. And because dosing, because there was really no precedent for a lot of this stuff a couple years ago, we're probably actually low on the dose, seeing what everybody presented here, where they're going in with 1 billion cells into the CNS. And we started with 10 million cells. I think this is a learning process. I think the characterization assays will get more standardized, though everybody's using something completely different, from embryonic, neural, mesenchymal, allogeneic, autologous, embryonic. So in a way, everybody has to come up with their own characterization assays for the specific cells that they're putting in. But I think because of all these different types of cells going to phase 1, it should get a little bit easier, a little bit calmer going in, especially since nobody's seeing any major immune responses so far, even on the allogeneic lines. So I think as we go into phase 2s, where we want to up the therapeutic efficacy, we have to watch what's happening with the other studies and learn as much as we can from once people start publishing their results from the phase 1 studies. Be important.
So Ellen, I mean, because it's interesting. So yes, you're right. There's different cells. People are using different tests for characterizations. And I guess from your perspective, how do you see the interactions with the agency helping companies to work through the challenges and just the differences that each project may have?
Yeah, I mean, I think there's a couple of different issues. One, there's just the scientific and the technical issues in terms of the different cell platforms, and it can range the gamut anywhere from the type of preclinical animal model that's looked at, and some of the different disease states really don't have a very well-defined animal model to use, so there's a lot of ambiguity about what they should be doing, and then even if they do it, what does it really mean for the human disease state, and how does it extrapolate? I think there's a lot of issues with the manufacturing, with the cell characterization, and the point at which you move towards that more GMP and commercial manufacturing. But I even think it starts even before that in terms of just the interactions with the FDA. I mean, one of the things we were trying to do with the paper, which is really talking about communicating with the agency, is the why, what, when, and how. And what kind of questions do you actually want to make sure that you actually put on the table, and how do you want to prepare so that you can actually have informed, productive discussion? Because at the end of the day, you want to be able to have informative and productive discussions with the agency. And part of it is the preparation for that and knowing what questions to ask and how to move through it. And the second is all the non-scientific, the tone of how you approach that communication. So I find with a lot of our investigators, some of whom are from academics, some of whom are from small companies, a minority are from actually companies who have a lot of experience dealing with the agency, is actually starting at the very beginning step with, we need to move down this pathway together. It could take several years. And so part of it is to really establish that credible type of informative, productive interaction. So part of what we try to do is think about what do you need to have answers to so that you can move on to that next step, and what are your milestones in the development pathway. So, it starts, at least for cell therapy, can start with that pre-IND, very informal discussion with the FDA, go through that pre-IND, and although I think it was mentioned earlier, they might have had 5 pre-IND meetings. That I'd say is highly unusual, and usually you get 1 pre-IND meeting with the agency. Perhaps you can have other types of ad hoc interactions in the interim, and then there's a pre-BLA and a post before that time, post-phase 1, post-phase 2. But there's very defined time points when you have interactions. And I think part of the challenge is making sure the investigator, the sponsor, is asking the right questions.
Okay. So we're still on challenges. We can.
Yes, we're still.
Yeah. So I mean, I know some of you have heard me talk about this before. I mean, I think one of the challenges and one of the issues for this space is what is the appropriate trial to do first. I think there's options, right? You can do an open label study. A lot of that's been done in cell therapy. You're going to get some safety, maybe feasibility, but very little efficacy from that. You can do a phase 1/2, smaller, maybe have a control arm, gives you a little bit more information, but certainly no definitive efficacy because you can't really get to the approval endpoints with that kind of a small kind of number usually. And then you could jump more into a phase 2 or some adaptive design. And I think clearly all of that's driven by the FDA's view of risk. Right, the risk of the therapy and what's going on. So maybe I can share what we did in one example, and that was in the stroke area that we just, the first study that we're doing. And we went in, this was a study that we're asking the FDA to do something that's never been done. We're saying we want to inject stem cells into the carotid artery 2 weeks after a stroke. Never been done in the U.S., had been done in Brazil safely, but never done in the U.S. So obviously the agency is going to be a little concerned about that, and their concern is that either the cells will aggregate and maybe potentially have another stroke, or that in going up into the carotid artery you may knock something loose and then create another stroke. So that, obviously I think they're legitimate concerns that we have to address. But we also knew that in this area we could not get any really valuable efficacy data by doing a phase 1/2 or an open label study. So we went in with the notion that we had to go do a sizable study so that we would know whether or not these cells actually worked. And so I think we compromised with the FDA where they asked us to do, we're doing a phase 2, but the first 10 patients in the phase 2 were done sequentially. And then at the end of 7 days, we had an independent DSMB which reviewed the safety data and basically signed off and allowed us to move on to the next patient. So we were allowed to do the first 10 patients in 3 sites, and then once that was cleared through that safety phase, we were able to enroll in 15 sites and then complete the rest of the enrollment. So I think it's an example, I think, where we had an objective. The FDA obviously had concerns, but we were able to work with them to put a structure in place, I think, that met all the needs of the parties.
So Patrick, from the later stage perspective, I know that your company, you had dealings with both CDRH and CBER. And so from an interaction perspective, what are some of the challenges, and from your future perspective, for companies coming into the space?
Well, I think, earlier stage companies, you tend to have, scientists who are, coming out of academia because they have a, specialty in the particular cell biology area or related area, and typically with not a lot of experience in the regulated environment. So I think that's an evolution for a company to be able to, you need to have the scientists, but you need to be able to communicate effectively with the agency. So I think early stage companies are probably going to need to bring in consultants to help guide the process, participate in meetings with the agency, until you have that skill set internally. But it's important to be able to effectively deal with the different groups. There's a number of different groups you're dealing with the agency at the preclinical, clinical, manufacturing, division of manufacturing, product quality. So it's a very diverse interaction with the agency, and you need to be able to speak the language of each of the different groups that you're dealing with. But we found the experience both with CDRH as well as the Office of Cell cell products to be, very interactive, very scientific based with both groups, and we find very open. At the end of the day, they have a job to do to assure the safety and effectiveness, but I think we found them to be very cooperative. But you need to go in, you have to be credible, you have to speak their language, and you need to prepare, as Ellen said, you need to go into meetings very well prepared and being able to present your case effectively.
I just want to make a point. In addition to these meetings, I know at our institute we have interactions with the agency where we put together educational webinars, which we post on our website, which are trying to tackle a particular challenge on the pathway, like the host immune response or preclinical animal models, or how to image or track cell fate. In addition, we have roundtables once a year where we bring together probably 15 or 18 people from the Office of Cell Tissue and Gene Therapy and people from academia and industry to focus on a particular topic. And the last one was on clinical trials. And there we got on the issue of, do you have to go into the most advanced, no options patient where you really don't expect to see anything that can really help you develop your product, but potentially the benefit risk is there that at least you're doing no harm, to what are the endpoints that you really want to look at, because we're looking at the traditional endpoints of clinical benefit, but then some people had actually brought up, since we're trying to repair and regenerate, are there other types of endpoints, maybe they're not yet validated, that maybe we as a field should start thinking about so that we can try and advance this field in a more accelerated fashion?
So I guess it really is very indication specific, though, when you're talking about clinical trials and what the agency is going to accept, what types of endpoints and patient population.
But they have, certainly common concerns for cell-based products: tumorigenicity, persistence. The biodistribution of cell-based products is obviously very unique and much different than a typical biologic. And how do you study that preclinically? It can be a real challenge. But so there are, I think, common themes for cell-based products, but obviously therapeutic area specific considerations.
Any unique issues that come up from, let's say, kind of from the early, like very early stage, from going from...
Yeah, making all the mistakes that I can make, and so for the second time around, I hope I won't do it. And that you don't have to say yes to everything the FDA brings up and asks you to do. They were kicking me under the table to stop volunteering all these preclinicals. So I can do that, I can do that. It's a negotiation. You have to be within reason. I'm not going to take CSF from 100 mice. And so they were more, I don't know if it was because we were academic rather than industry. They're a little bit, there was a lot of discussion and consideration back and forth. Sometimes you get a new reviewer on the panel that brings up something that wasn't brought up till the last minute. For us, it was, and we went in wanting to do multiple rounds of treatment and this and that, and they cut it to bare bones. So I think initially, to get your foot in the door, you make it as conservative and as a safety study. But we saw as soon as we finished the first cohort, we can put an amendment to open it up a little bit more, and then after the second cohort, another amendment. So for us, the strategy with the clinical PIs anyway was to go in conservative, don't rock the boat too much, get it in, get your cell product in, learn what you can from the first cohort, and then put amendments in to, and hopefully that process will speed up along the way, that we don't have to do 12 patients now to move to the next thing, that 3 or 4 showing safety will let us get to the next step faster.
But you found that once you started getting patients in, then, so would the interaction with the agency just be via telephone, email, or?
Well, we at City of Hope, they have a regulatory committee that they contact directly, so there could be emails and stuff in between, rather than wait till we finish our tox study and then they say, oh, by the way. So they do want the interaction, so we try to clear, especially IND-enabling studies, submit it, let them review it, get all their input before we do anything that's going to take a lot of time and effort. That's it. So as I say, it's sort of a learning curve for them too. So to go in with your arguments of being persuasive, so you're the expert going in with your product. So keep you can keep the upper hands a little bit. Yeah, it's a lot.
And so, Tim, because I know you deal with various clients, is there any issues that you see that come out, whether it be from the early stage, prior to phase 1, just starting out, then going into later stages of manufacturing?
Yeah, there's, I guess in our experience, one common theme is again, going back to the product characterization assays. Many of the clients come to us, again, coming out of the university or academic setting in which the process isn't well defined, so it's our job to define that manufacturing process, make it GMP, try to remove as much manual labor as possible, and go from perhaps handling half a dozen or 20 flasks to maybe a larger bioreactor. So all these types of process changes, I think, from a regulatory perspective, is you have to take a step back and try to understand the science and the cells and what you're trying to do to these cells. Is going from 24 flasks to a large bioreactor a significant change in your process? Probably not. But if you change your media, or you change your supplements, or you change how long you culture your cells and expand them, then perhaps that is a major change. So I think from a regulatory perspective, it's important to have these cell characterization assays, maybe not optimized or perfected, but at least have a few in mind, so that as you make these process changes, you can go back to the regulatory agency and state your rationale, state your case, and show data to prove that whatever changes you've done in your process to make it bigger, to scale it up, to scale it out, to improve yields, to increase your expansion 50-fold, that you really haven't changed the fundamental aspects of your cell, not only from a phenotypic point of view or a molecular point of view, but more importantly from an activity point of view. So again, I think, at least for us, that's been a common area where a lot of clients come to us and say, yeah, I've done a FACS analysis with these 3 markers and that's what I've got. Well, we'll help them through the process and think a little bit more about what exactly you really need to know about your cells. So as you go through the whole development process, that the final product you end up with in clinical manufacturing and commercial manufacturing is really what the idea and concept was early on in the academic lab.
I just want to say also, what I've learned is also coming from academics, like it's a big deal just to get it through into the phase 1. You're really not thinking about phase 2 and commercialization. And actually through conversations with SCMOM, the earlier we get input from that, it really changes your phase 1 manufacture, production, how you design the trial. Because what we could do for a phase 1, we can't do for a commercial
Exactly.
project. So to try to get the input earlier from the end stage back early. So that's something we learned for our second product to get consultants earlier on.
Yeah, I think that's an important concept, and I think from this morning's earlier panel discussion is that one thing I mentioned is that we don't expect the manufacturing process for your phase 1 clinical trial to be the process that you take all the way to commercialization, because manufacturing for 24 patients a year versus 10,000 patients a year is going to be tremendously different from a materials management point of view, from a manual labor point of view, from product characterization and release testing point of view. Just the types of validation you have to go through for all these assays and processes. So, clearly I think it's a dynamic process where it's good enough for phase 1, and you've got some aspects or some ideas about what your product looks like from a phenotype or a molecular or a in vitro activity point of view. And it's, as you go through phase 1, phase 2, then you begin to refine those and tighten down some of those specifications until you get to your phase 3 in commercial manufacturing. Again, as long as you've got all the data throughout the whole process, you can justify back to the regulatory agencies that what changes you've done from phase 1 to phase 3 in commercial are justified and that your product is still essentially the same thing.
Yeah, so that might be some of the challenges that companies face there, because you're right. It is a stepwise approach, and you're learning about your product. You're gaining information on your clinical program. But, companies need to be aware of what you need to do, but whether it be from comparability perspective, if you do change your process. And I could see where that could be a challenge if you use something in your IND enabling your preclinical tox studies, one lot of material, and then something was changed, and now you want to go into phase 2 or further phases. Any input on companies in regards to how to address that particular challenge?
Well, I think it's along that line. It's very important to have a very strong comparability panel or bioequivalence panel, so that when you do make process changes, or for an allogeneic product, when you introduce new cell strains, as we do several times a year now for our Apligraf product, and we've had something like 60 process changes over the years. A lot of them are to decrease the cost of goods, just commercial realities. You need to make the process less expensive, removing animal components from media, other changes to prevent risk of contamination in the process. So each time you make a change like that, you want to have a comparability panel, so that obviously in an extreme case you don't want to be locked into generating initial clinical data. That's obviously can be a huge setback. But if you have a characterization panel, a comparability panel, as we do, that's both in vitro, in vivo functional assays, I think that can prevent a more extreme situation.
And so Ed, you spoke earlier about you've actually used several opportunities, whether it be from special protocol assessments within your programs. How did those opportunities come up? I mean, did you, from a strategic perspective within the company, say we want to utilize these opportunities, or did the agency offer them to you? How did that work out?
I mean, actually, it follows on a comment that Ellen made about composite endpoints. So a little bit of background. We did a phase 1/2 study in critical limb ischemia. And, for those people who know the space, the only endpoint the FDA would ever accept for approval is amputation-free survival, which is a very difficult endpoint to hit, right? Lots of patients. We talked to a lot of thought leaders in the space that came up with a composite endpoint that included AFS as well as other measures. And we thought, let's go to the agency under an SPA and see if we can get that accepted. We very quickly found out that they would not accept that. And I think it's one of the lessons of, I think it's neat to think about composite endpoints. It's great. It reduces the size of the trials. That's wonderful. But it takes a lot of work to validate those. And if you want to see how fast an investor can run, tell them that you're going into a phase 3 with an unvalidated endpoint as your primary endpoint, because they will run faster than you could ever imagine. So, I think we play that, thing, but we wanted to test the composite endpoint. That's why we went to the SPA route. And, it's not a quick process either. I mean, it took about 6 months to kind of get through all the discussions about the composite endpoint until they finally just said, we're not going to accept it. And we went back with a study that had amputation-free survival, and they signed off immediately on it. So, I think it's a process that you initiate as a company, because you want that definitive answer about what that study is going to look like. But it's not a quick process, and it does take quite a bit of effort.
But I think what you're saying is, don't invest in a pivotal registrational trial with an unvalidated endpoint unless you've had a discussion and have buy-in with the agency that that's going to be an approvable endpoint.
Well, and even, I'd argue not even go to a pivotal. You have to go do a phase 3 with a composite endpoint to get enough data to say that this may actually work, right? And then, again, back to raising money to do that is darn near impossible, right? Because there's so much risk in that kind of a step, right? And, people have made these arguments about cell therapy. Oh, well, they're special cells and they're stem cells and they do great things. We shouldn't have to, meet the endpoints that small molecules meet, because these are great therapies. But, the FDA doesn't view it that way. I mean, there are set endpoints for these diseases, and it's whether small molecule, protein, stem cells, you have to hit them. So we don't get any shortcuts from that perspective.
No, the only, not really a shortcut, but some measure of biologic activity that you might cause the company to think, okay, this is encouraging enough, I'm going to take that next step, as opposed to some people do mini phase 3s as a phase 2, and so there might be some sort of biomarker that you can try and get it, a little bit of data to support it. For example, in cardiovascular, I know people are talking about other endpoints other than ejection fraction, for example, which is a not a great, but it's a surrogate for a clinical endpoint. So people are trying to look at myocardial viability or other types of measures or scar mass. And so the issue is always show me the data.
Right.
And show me how that measure, even if you can objectively measure it, means anything in terms of a clinical endpoint of interest.
Yeah, so I mean, that's an excellent point, but even for the biomarkers, it is show me the data because the agency still wants to see it validated before they would say that they're accepting of your biomarker. I mean, some biomarkers, you're right, are already, for certain indications, they're already validated. You can use them, you can, use them for the health authorities to say, okay, this is viable, we can move forward. But in some cases where you're trying to, be innovative, think outside the box, it takes a little bit longer, and you're leading the way. So, are there any questions from the audience? I don't think so. No, quiet group.
Hard to see, right?
It's
Yeah.
very hard to see here.
I'll make another, I think, we've talked about the therapy side. I'll make a comment about the device side, because I think some people in the audience are thinking about device side. I've only been exposed to this over the last 6 to 9 months, but I think clearly the FDA allowed some point-of-care devices to get out in the marketplace under 510K that they wish they probably hadn't done. And they've seen this proliferation of use in a number of indications where these cells or other agents could be used. And I think it's caused them to take a step back and think about how they want to regulate these. And I think a lot of the discussions they're having with Cytori is related to that. We're trying to get an additional indication for our device that's been difficult just because, the cat's out of the bag, but they don't want to have more cats running around, basically. And I think that has some significant ramifications where they're pushing companies away from the 510K path to more of a PMA path, which is obviously much more expensive, takes a lot more time. So I think even the device side of this business from a regulatory perspective is evolving.
So what are some, I ask each of the panelists, what are, I guess, your advice in regards to the do's and don'ts of interacting with the agency to try to assure regulatory success at each stage of your development? And it's a broad question, so you can take with it what you will.
You're going to start at that end?
Well, sure, I'll take the first stab. So I think, at least in my experience, it's the FDA, the folks at the FDA are reasonable. I think again, as I think the panelists said, be prepared. Make sure you have a good scientific rationale for what you're proposing. And really, I think at least what I found, at least in the cell therapy field, that not only the FDA, but also the MHR, EMA, and other agencies worldwide, they realize this is a new field. They realize regulations and guidelines are evolving, and they're willing to negotiate. And I think if you present a rational, scientific, technical-based argument for what you propose, then more often than not, they will allow you to do that. Again, with limitations, and perhaps with the caveat that as you move forward from phase 1 to phase 2, that you tighten up testing or specifications or whatever else in your process. But approach it that our approach has been, at least when we work with our clients, is it's a negotiation. Just be prepared, and I think things will work out.
You're next.
I mean, I think, what you said is correct. I mean, it's evolving there. I think they're flexible if you make good scientific, rational arguments. I think it's also interesting, I think there's a learning curve with each technology. We saw it, our first IND took, I think, 120 days to get clear. The next one took 90, the next one took 30, the next one took 30. So, I mean, as they get more comfortable with the technology that you're going forward with, I think that the process improves. They understand it. So you have to expect that there's that learning curve. And our experiences have been, if we have good arguments, good data, that they're usually pretty flexible in terms of, coming to a compromise that meets, both parties' needs.
Yeah, I mean, I would agree with that. Certainly, even through a 10-month BLA process, we saw the learning curve with the reviewers that we were interacting with. And I think there's also because this is a very new field and there's a lot of new restrictions or considerations with cell-based products. I think it's important. I mean, obviously, FDA doesn't really have a concern about business type issues, but they do need to be aware of the impact of some limitations of cell-based products like shelf life and introduction of new cell strains and things like that that really do have practical limitations on the manufacture, distribution of cell-based products. So we found them to be very receptive to that, once, again, educated on what the issues are, what the real-world limitations are. So I think they've shown a lot of flexibility to adapting to the unique considerations or restrictions of cell-based products.
I don't have much to add. I think CBER must be overwhelmed right now with all new applications going in. It does take time. I think even for pre-IND, they're asking for 60 days in advance. So,
Yeah, they are.
Yeah.
so just to get it in and leave yourself time.
Well, and I think just to go to that point, CBER actually, the division really does take the time to meet with companies. So if you want to speak with them, you might not be face-to-face, but they do take the time to have telecons and make sure that they interact with you if you actually ask for time. So I think that's what is also keeping them very busy, Karen, besides the number of INDs that are probably being submitted.
And I think having seen, at CERN we have about 75, we have 480 awards, about 75 translational projects and about 25 different teams. So we've seen, autologous, allogeneic, a variety of tissue engineered products. So there's a variety of things that are going through the system right now. But I'd say stay, message-oriented. You know what you're trying to ask and get across. Be clear with your thinking. Be prepared as you go in there, and give them a hint of where you're trying to go with your target product profile. Eventually, even if it's aspirational, it always helps them to know where you're headed, because then they're probably going to be more likely to be able to help you. It's actually a science-oriented discussion. It's a data-driven discussion. They don't like hypotheticals. Mm-hmm. So if you pose a hypothetical, they'll come back and say, it depends. So, they really do want to see the data and the specific issues that you want to have discussed. And if you're asking a question, they're not your co-developer, they're a regulator. So the point is to ask the question you need to move your product forward, and make sure that you prepare a package of information that could actually inform the discussion. If you just ask an open-ended question, is this okay? That probably won't fly very well, but if you say, I want to do this because, do you agree? They're more likely to come back with an answer that might be helpful to you. So I think from our point of view as an institute, and since this is a new technology, and since for many of these sponsors, they're new to the agency and new to product development, we're in favor of over-communicating. And, there's a lot of different places where you can communicate with the agency, so take advantage of it. I'm not worried about them being overworked. I'm more concerned that you get your questions answered. And so I think that's what we would say is if you have an issue, try and be as transparent as you can be, and also be aware if you ask a question and they give you an answer. My point is, be careful of what question you're asking, because they give you an answer, you're going to have to deal with that response and have to figure out how to deal with it.
Yeah, I think for any type of meeting, the preparation of the briefing package is critical, because that's what educates them on the product, clinical trial, preclinical, whatever you're going to the meeting for. And they don't like to have open discussions. They want to specifically respond to something concrete, and it's really got to be all set forth in the briefing package. So the background, and then you end the briefing package with specific questions, and that's what they're going to want to respond to in the particular type of meeting. They just don't want to have open discussions necessarily. The point of the meeting really is to get clarity, clarification around the questions, but generally, what you present in the briefing package is what's going to be discussed at the meeting. So you need to spend a lot of time preparing a well thought out and very specific briefing package.
and making sure that your questions are questions that you need the answers to.
Yeah.
Right? So, I mean, that actually is a very important statement that you said.
The other thing, I think, is just if, I hate to use the analogy, there's a dead skunk in the middle of the road, but it's better that you find it than they find it. So if there's a problem, it won't go away. So, you know, if it's a significant problem, it's probably better that you identify it and bring it up and propose how you're going to address it than hope nobody sees it, because that probably won't go over very well. So those are just some things. You have to be careful of what question you ask, but if there's a real challenging issue there, it's probably better to come very prepared, identify it, and let them know how you're going to approach it.
So any questions from the audience? Yes?
Could you speak a little bit to the harmonization around the globe of regulatory requirements and where there's most likely to be discrepancies for developers?
Do you want to address that one? Well, within ARM, we've had some discussion on some of those issues, and I don't know if Kathy
Yeah, so-
wants to go into
So
that.
that's actually a very good question. We could have a whole panel on that ourselves. And so within ARM, the regulatory committee, that's one of the workshops that we are looking to have early next year, because there are certain areas specifically within the quality or the CMC aspects where we need to make sure that both the EMEA or the European guidances and regulations can match up with with the U.S. ones. And so I don't know, from a quality perspective, Tim, if you have any insights on that.
Well, I have a disclaimer. I'm not a card-carrying regulatory person, so. But no, I think, at least, again, from my perspective, I see a general trend of harmonization, both from the regulatory guidelines as well as from the quality perspective. So, again, you know, my view may be limited and fairly narrow, but from a manufacturing point of view and a product point of view, I think agencies around the world are working towards the same thing.
But I do think those discussions are happening, and ARM is trying to organize a meeting to talk about which aspects are common across the different continents, and there certainly are some unique differences, and since companies, many of them are global, we do want to have a meeting of the minds so that at least people are reading from the same book, so that when they're designing a study, they're thinking of not just the United States, which is of course one of our major foci, but they're also thinking about, you know, what do they need to do to make this a global product. So that is a workshop that is in the works right now.
I'll make one other comment about that. There's one side, which is the positive harmonization, right, of regulation. There's the reverse harmonization of unregulated activity abroad that I think has permeated some of that activity in the United States. So it's coming kind of reverse way instead of the, together. And I think that is something that is affecting all of us and has the potential to affect all of us in a great way. And I think that's something that ARM has taken on as well, with the code of conduct that all members of the organization apply to the regulations in the areas in which they conduct their business. But that kind of reverse harmonization, or however you want to phrase that, I think is a real issue for this industry.
Yeah, I think we're finding in our space that there's a proliferation of tissue products that are coming onto the market, basically self-designated as a 361 HCTP product. And if you're familiar with the human tissue regulations, a 361 HCTP can go on the market simply complying with good tissue practices if it's for, if it's homologous use, minimally manipulated, and the function of the product is not related to the biochemical or metabolic activity of cells. But that's, in essence, unless FDA designates a company to go before the tissue reference group at FDA, it's essentially at this point a self-designating process. So we're seeing a number of tissue-based products that are coming onto the market and making claims related to the cells or cytokines growth factors produced by those cells where they don't have valid scientific evidence to demonstrate safety, efficacy, or the effect of the product. And I think it's a big threat to the companies within ARM who go through the regulated process to demonstrate valid clinical data for safety and efficacy. And one of the problems is when these products get onto the market, the clinicians, they don't understand the regulatory frameworks, they don't understand how products get on the market. So they can be using products on patients where claims are made, but the claims are not substantiated with valid scientific evidence. So it's something that ARM is looking at, and we believe it's an important issue. It's not as out there as the stem cell tourism issue, but it's a real issue of maybe lack of regulation of a certain class of tissue-based products.
And one thing I'd just like to say from the sermon is we're actually having a small workshop November 14th and 15th on what we're calling the Alpha Clinic concept, because we do have, really a pipeline of products that are approaching or about to go into the clinic. And so we are trying to think about what's the capacity, what are the resources, what are the issues that we need to be thinking of to prepare for the future, so that hopefully we can steer people to, qualified clinics or reputable places where people can go for their clinical trials. So perhaps we can decrease some of the pressure to try some of these very high-risk business operations rather than something that's actually going to be helpful for patients. So we are trying to figure out a way to put something like that together as a resource.
Was there one more question?
Think of the regulatory process as a whole. The end goal is to get an approved product. Much of the discussion has been around the ways of positioning your product in order to do studies and whatnot. Does the panel feel that the ultimate test to get the approval to get on the market is going to be sort of the result of the sum total of the studies done before, as you would do a small molecule or a biologic, or do you think that there's going to be a higher hurdle before the agency actually takes the plunge and approves these products for market?
Well, I just made a comment, one size doesn't fit all. So we run the gamut from human embryonic stem cells to adult stem cells. So I would say for the very early derived, there's going to be some long-term safety issues. I don't think they're going to demand that up front. I think that's going to be probably a post-marketing commitment in terms of having some sort of a registry or some sort of a long-term follow-up for those types of cells. For cells that don't survive that long or other issues, I don't see that being a part of the mix. But those are just some initial thoughts.
I'd say our conversations with the FDA have said that we don't have to achieve anything above what a small molecule would be in order to show efficacy in a given indication. So there's been no indication that we'd have to go beyond what you'd have to show with a small molecule or a protein or antibody.
Well, then, thank you very much, panel. Thank you all.
Thank you.